All right, hello. We are back with another science session. One second, let me just check my mic. With another. Okay, it works. So, yes, let's start off with what is a buffer. So, a buffer is a solution that will resist changes in pH when small amounts of acid or base are added. So, it's a solution that resists changes in pH when a small amount of acid or base is added, and it usually consists of a weak acid and a conjugate base, or a weak base and its conjugate acid, and it helps maintain a stable pH in the biological systems. For example, our blood uses the bicarbonate buffer system. So if I ask you, what's a buffer? A buffer is a solution that that resists changes in pH when small amounts of acid or base is added. And what is an action potential? So an action potential, it's a rapid temporary change in a cell membrane's potential that occurs when a neuron or a muscle cell is stimulated, and it involves depolarization followed by repolarization, allowing the electrical signals to propagate along neurons or muscle fibers. So what is an action potential? It is a rapid temporary change in a cell membrane's potential. It it happens when a neuron or a muscle cell is stimulated. Again, an action potential is a rapid temporary change in a cell membrane's potential, and it happens when a neuron or a muscle cell is stimulated, and it would involve depolarization. That's when there is an influx of sodium or Na plus. And then after the influx of sodium or depolarization, there could be a plateau, and there's repolarization because there's the efflux of potassium. It is going out, and that will allow electrical signals to propagate along neurons or muscle cells or muscle fibers. So what is DNA? It is deoxyribonucleic acid. Deoxy. Deoxy. That's why Deoxys from Pokemon is red and blue. Okay, Deoxys. It is the molecule that carries genetic information in living organisms, and it is composed of four nucleotide bases: A, T, C, and G. Adenine, thymine, sine, and guanine. It is arranged in a double helix structure and encodes the instructions for building proteins and regulating our cell function. Okay, so it carries genetic information in living organisms. What is DNA? It carries genetic information that in living organisms, and it's composed of four nucleotide bases ATCG arranged in a double helix structure. Regarding A, T, C, and G, A to T, adenine, two bonds, to thymine, and C3G, it's cytosine, three bonds to guanine. But what is RNA? It's different from DNA. So RNA, or ribonucleic acid, it's single-stranded. It's a single-stranded nucleic acid in gene expression, and it comes in very varied forms. So it can also carry the genetic information from DNA to ribosome. So for example, several forms of RNA is mRNA, which would carry the genetic information from DNA to ribosome. tRNA, brings amino acid during the translation, and rRNA forms the structural and catalytic core of the ribosome. So far, we did 4. So what's a buffer? A buffer is a solution that not temporarily, it would resist the changes in the pH. And it could be when you add acid or base. Also, a buffer can be a weak acid and its conjugate base, or a weak base and its conjugate acid, and it helps maintain the stable pH in different biological systems. What's an action potential? An action potential is a a small change or a temporary change in the membrane potential, and it happens when a muscle or a neuron is stimulated, and it involves depolarization, aka influx of sodium, and also the efflux. Oh, after that one, there's repolarization, and there's the efflux of potassium, and it would allow electrical signals to be propagated along the neurons or muscle fibers. And what is DNA? It carries the electrical signal or genetic information in living organisms, and it's composed of 4 nucleotide bases: A, T, C, and G, adenine, 2 bonds to thymine, cytosine, 3 bonds to guanine, and it is arranged in a double helix structure. And DNA encodes instructions for building proteins and regulating cellular functions. RNA is similar but different. DNA. Hello. Ooh, wait. Yes, I might miss some chats because I'm focusing, but hi, thank you for dropping by. RNA is ribonucleic acid, and it is a single-stranded nucleic acid involved in what is it involved? Involved in genetic information. Next is, what is transcription? It is the process by which RNA polymerase synthesizes RNA from. Again, transcription is the process by which you make, oh, let's use some fancy terms. Transcription is the process by which RNA polymerase synthesizes RNA from a DNA template. Okay, it involves initiation at a promoter, the elongation of the RNA strand, and termination once the signal is reached. Okay. So what is transcription again? It is the process by which RNA polymerase will synthesize RNA from a DNA strand. So coming from a DNA strand, you make RNA, and it starts at initiation of the promoter, and then elongation, and then termination once stop signal is. What is a gene? Okay, we have to make sure that we don't mix up everything. A gene is a sequence or a specific sequence of DNA that would encode a functional product, and it's usually the product is a protein or a functional RNA, and it includes regulatory regions like promoters and enhancers, in addition to the coding sequence. So what is a gene? A gene is a sequence of DNA that encodes a functional product, and that is usually a protein or functional RNA. Again, what is a gene? A gene is a specific region in DNA which would encode a specific product, could be RNA or a protein. And it would include enhancers or silencers. Enhancers, promoters, coding a sequence. And what is the central dogma? Central dogma of molecular biology. Well, that describes the flow of genetic information. So, DNA is transcribed into RNA, RNA is translated into protein. Remember that the central dogma describes the flow of genetic information in human body generally. So DNA is transcribed into RNA, RNA is translated into protein. What is the central dogma? The central dogma describes the flow of genetic information. DNA is transcribed into RNA, and RNA is translated into protein. What is an enzyme? What is an enzyme? An enzyme is a catalyst that speeds up chemical reactions without being consumed. So again, an enzyme is a biological catalyst that speeds up reactions without being consumed. It would lower the activation energy of a reaction. And sometimes or often it shows substrate specificity. So an enzyme is a biological catalyst that speeds up chemical reactions without being consumed, and it would lower the activation energy of a reaction without being consumed. Yeah, it shows substrate specificity. And what is ATP and why is it important? You always hear ATP. The ATP or adenosine triphosphate. It's the primary energy currency of the cell. It is the money. It's the primary energy currency of the cell. It stores energy in its phosphate bonds and fuels the cellular processes such as transport, metabolism, and signaling. So it is the primary. So what is ATP? ATP or adenosine triphosphate is the primary energy currency of the cell. It stores energy in its phosphate bonds, and it would help with different biological processes. That one. What is a protein? A protein is a polymer of amino acids linked by peptide bonds. A protein is a polymer of amino acids linked by different peptide bonds, and its function depends on its 3D structure, which is determined by the amino sequence. So the function of a protein depends on its 3D structure, which is determined by its amino acid sequence. A protein performs structural, catalytic, regulatory, and transport roles in the cell. Okay, let's go back to the other stuff that I said earlier. So what is a buffer? A buffer is a solution that would resist changes in the pH. It could be when you add an acid or a base, and also it is composed of a weak acid and its conjugate base, or a weak base and its conjugate acid. And an example of this is how the blood uses the bicarbonate system to maintain a stable pH. Next, what is an action potential? An action potential is a rapid but temporary change in the membrane potential of a system or a cell, and it occurs when a neuron or a muscle cell is stimulated. And the way it happens, or it what is involved in an action potential? Well, there is depolarization when sodium influx happens, then there it is followed by a plateau, but there's also the repolarization where potassium efflux happens. What is DNA? It is genetic. It carries the genetic information, and that is deoxyribonucleic acid. Also, it is composed of 4 bases: the adenine, thymine, cytosine, guanine, adenine, connected using 2 bonds to thymine, cytosine, 3 bonds to guanine. What is RNA? What's its difference with DNA? RNA also carries some genetic information, but it is single-stranded compared to DNA, which is double-stranded, arranged in a double helix structure. So RNA is a single-stranded nucleic acid. Can be in different forms. There's mRNA. An example is mRNA, tRNA, and rRNA. mRNA carries genetic information from the DNA to the ribosome. tRNA would carry the codons or brings amino acid during translation, and rRNA forms the structural and catalytic core of the ribosome. And what is transcription? It is the process by which RNA polymerase synthesizes RNA from a DNA template. Again, what is transcription? It is the process by which you go from DNA to RNA. You synthesize RNA. We can talk about the very specific steps later. tRNA, wait, no, that's not it. Next, transcription, again, DNA to RNA. It involves initiation at the promoter, elongation of the RNA, and then termination at the stop codon, stop signal. What is a gene? A gene is a very specific area in a DNA, a very specific sequence in DNA which encodes product, functional product, which could be a protein or a functional RNA, and it would include regions of motors. What is the central dogma of molecular biology? It is it dictates how the flow of genetic information from DNA. DNA is transcribed into RNA. RNA is translated into protein. What is an enzyme? An enzyme is a biological. It's a catalyst. It's a biological catalyst which would help reactions go faster. And the way it does this is by lowering the, what was that, EA? The activation energy. It lowers the activation energy and is it's specific to substrates. What is ATP? ATP is, what is ATP? ATP is like the money, is the primary energy currency of the cell, and it stores energy in its phosphate bonds, and it fuels cellular processes such as transport. All right. Hello. Yeah. I storm that is approaching. Okay. It's all right. Next is, what is a protein? It is a polymer of amino acids linked by peptide bonds. There's amino acids linked by peptide bonds, and it depends on its 3D structure, which is determined by amino sequence. Okay. Amino acids linked by peptide bonds, that is a protein. And its structure depends on its 3D structure. What is a neuron? A neuron is a specialized cell that transmits electrical signals in the nervous system. A neuron is a specialized cell that transmits signals in the nervous system. Consists of a cell body, and the dendrites would receive the input, and the axon transmits the input. Again, a neuron is a specialized cell in the central nervous system, or in the nervous system, and it is composed of a cell body, and where the dendrites receive the signal, the axons would send out the signal. What is depolarization? It is a shift in the membrane potential towards a more positive value or a less negative value, usually because of the influx of sodium ions, Na+, during the initial phase of an action potential. What is the sodium-potassium pump? Important. It is an active transport protein that uses ATP to move 3 sodium, 3Na out, 3Na out, and 2K plus in. What is the sodium potassium pump? It is an active transport protein that uses ATP to move 3 Na ions out and 2 potassiums in. And that would help maintain the resting membrane potential and ionic gradients. What is the nucleus? The nucleus is a membrane-bound organelle that houses the cell's genetic material. And what was the genetic or the material? It is the DNA. So where is the DNA? Where is its address? It is in the nucleus. It contains DNA, which is organized as chromatin, and it is also the site of transcription and RNA processing. So the nucleus is a membrane-bound organelle where the DNA resides, and the DNA is the genetic material, and that is also where transcription happens. Remember, transcription is from DNA to RNA, and also where RNA processing happens. We will talk about that later on. Next, what is the mitochondrion? It is the powerhouse of the cell. It generates ATP via oxidative phosphorylation, and it also plays roles in apoptosis, calcium homeostasis, and metabolism. So what is the mitochondrion? It is the powerhouse of the cell. It generates ATP via oxidative phosphorylation. And what is the endoplasmic reticulum, the ER? It is a membranous organelle involved in protein and lipid synthesis. The rough ER has ribosomes for protein synthesis, while the smooth ER is involved in lipid production and detoxification. So, what is the ER? It is a membrane organelle involved in protein and lipid synthesis. The rough ER and the smooth ER. Rough ER is for, it has ribosomes for protein synthesis, and smooth ER is for lipid production and detoxification. Next, next we go and discuss some nervous system and membrane potential details. What is the resting membrane potential? The resting membrane potential is the electrical difference or potential difference across the plasma membrane of a cell at rest. So the resting membrane potential is the electrical potential difference across the plasma membrane of a cell at rest. It's usually negative 70 MV in neurons, and it's maintained by the sodium potassium ATPase pump. Mentioned that earlier. 3 sodium out, 2 potassium in, and also by lead channels. Especially using the potassium channels that allow potassium to diffuse out of the cell. So that would create a net negative charge inside. Of course, because if you allow potassium to just freely diffuse out, given how it is K plus, the pluses are going out. So what's left? Less plus, less positive, aka kinda negative. So again, what is the resting membrane potential? It is the electrical potential difference across the plasma membrane of a cell at rest, typically around negative 70 MV. How does the action potential propagate down an axon? So when a threshold potential is reached, voltage-gated potassium channels open, causing the potassium influx and depolarization because they can come in. And that local depolarization triggers the adjacent sodium channels to open, and that would propagate the signal. Afterwards, potassium channels open, allowing the K plus to exit the cell, which would repolarize the membrane. And this continues in a wave down the axon. Again, so how does an action potential propagate down an axon? So when a threshold is reached, sodium voltage gated sodium channels open, and that would allow sodium voltage gated. Again, let's start. So when a threshold is reached, voltage gated sodium channels open, sodium Na voltage gated sodium channels open, and that would allow the sodium to rush out. All the NAs are rushing out, and that the Na plus, it's plus. That would mean that the inside becomes less. Wait, what? Wait, one moment. The directions, I'm confusing them. When a threshold potential is reached, voltage gated sodium channels open. It's influx. Sorry. I was like, I know it's depolarization. Okay. So it's influx. Again, from the beginning. When a threshold potential is reached, sodium gated, voltage gated sodium channels would open. And that would allow Na plus to come in. A lot of them. The pluses are coming in. Therefore, the inside is more plus now. And that means it is depolarized. Less negative. Alright, we got that. And this local depolarization would trigger the adjacent sodium channels to also open, and that would lead to the other sodium Na plus to come in also, and that would propagate the signal. Afterwards, after it reaches a certain number, the potassium or K plus channels will open, and the K plus would then go out. So now that would repolarize the membrane, because depolarization earlier made the inside more positive because the Na pluses were coming in. But now that the K pluses are going out, there is less plus inside, therefore it is less positive, aka a bit more negative, aka repolarized. And this would continue in a wave down an axon. So why doesn't it go backward? Why doesn't the action potential go backward? Well, it's because of the refractory period. After a segment of the action fires, the sodium channels mentioned earlier would enter an inactive state and will not reopen immediately. So that's why it's only in one direction. It enforces unidirectional propagation. Again, why doesn't the action potential go backward? It is because of the refractory period. Yes, it is info. Because of the refractory period where the sodium channels enter an inactive state and cannot reopen immediately. And what would happen if the sodium potassium pump was inhibited for a long time? The ion gradients would dissipate, and that would lead to depolarization of the resting membrane potential, and this would impair excitability and could lead to cell swelling and eventual cell death due to osmotic balance. Because what's the role of the sodium potassium pump? It would pump the 3 sodiums out and 2 K plus in to reset the membrane potential. Next is, what is a promoter? A promoter is a DNA sequence upstream of a gene. Again, what is a promoter? It's a DNA sequence upstream. Usually in pictures, it's at the left side, upstream of a gene, and it would recruit RNA polymerase and transcription factors to initiate transcription. Again, transcription. DNA is transcribed into RNA. So what is a promoter? It's a DNA sequence upstream of a gene, which recruits RNA pol and also transcription factors. In prokaryotes, promoters typically contain negative 10 or the tata, and also at the negative 35, the titi jaka elements. And in eukaryotes, you have the tata box, and that also would generally require transcription factors for RNA pol 2 recruitment. Okay, what is a promoter? It's a DNA sequence upstream of a gene, and it is involved in the initiation of transcription. And also, there's a little bit of a difference between prokaryotes and eukaryotes. Prokaryotes would have the tata as a -10, and then at -35 it's a tt jaka. In eukaryotes, it has the tata box and requires different transcription factors such as TF2D for RNA Pol 2 recruitment. What am I talking about? Well, so regarding, I can walk you through the transcription initiation in eukaryotes. So transcription factors such as the TF2D bind the tata box, bind to the tata box in the promoter mentioned earlier, and then there is the assembly of the pre-initiation complex with RNA Pol 2. Okay, so TF2D binds to the tata box, and then there is assembly of the pre-initiation complex with RNA Pol 2. TF2H unwinds the DNA. That's the is no. TF2H unwinds the DNA. Isn't that it? Is that the same? One moment, cross-checking something. What is TF2H? Is that the same name? No, it's TF2H transcription factor 2H. Okay, but is that different from helicase? Oh, it is! It's a DNA helicase that opens the DNA double. So that's why. Oh my goodness. Transcription factor 2H. It is helicase. Unwinds the DNA. It is helicase. I thought they had a different name. Let me just write it. Okay, so transcription factors, just TF2D bind to the tata box, followed by the assembly of the pre-initiation complex, and then TF2H would unwind the DNA, and then RNA Pol 2 begins the RNA synthesis. After that, also phosphorylation of the ETD tail of RNA Pol 2 allows promoter clearance and transition to elongation. Okay, again. So in initiation, TF2D2 binds to the tata box in the promoter, and then pre-initiation complex is assembled with RNA Pol 2, and then TF2D or TF2H or helicase unwinds the DNA, and then RNA Pol 2 would begin the RNA synthesis at around +1. And how does it actually go from initiation to elongation? Well, you phosphorylate a certain tail, the CTD tail of RNA Pol 2. It would then allow the promoter clearance and go from initiation to elongation. How is RNA processed after transcription? Well, RNA is processed. Well, there's the 5' capping, splicing, and 3. It's the poly-A tail, polyadenylation. So pre-mRNA, you add 7-methylguanosine to the 5' end. Let's look at it this way: left, middle, and right. 5' capping, you add the 7-mg, 7-methylguanosine. Yeah, 7-methylguanosine to the 5' end. Middle, you splice. Introns are removed and exons are joined by the spliceosome. So the spliceosome will only keep the exons. We keep exons. Introns are removed. Next, the right, 3' polyadenylation. 5' cap, poly-A tail, 3' polyadenylation. You add a poly-A tail after the cleavage near the polyadenylation signal. What is the signal? It is a u a. It might depend, I don't know, different species, but that's what's written here. Again, how is RNA processed after transcription? 5' capping, splicing, and 3' poly A tail. 5' capping, you add 7-methyl guanosine to the 5' end. Splicing via spliceosome, you keep the exons and then you remove the introns. 3' polyadenylation, you add a poly A tail to the 3' end. And what's the 3' end? It is, there's a signal there, the AAU, AAA. Why is the 5' cap important? Yeah, why are we putting a 7-methyl guanosine as a 5' cap in the area? It would protect the mRNA from exonucleases. And it would help in nuclear export, and it's required for ribosome recognition and translation initiation. Again, by adding the 5' cap, the ribosome will then recognize it for translation. Also, it protects the mRNA from endonucleases, because without the cap, exonucleases, without the cap, the mRNA would just be degraded and inefficiently translated. Also, next is, what's the difference between transcription and replication in terms of enzymes and product? Okay, so transcription uses RNA polymerase to transcribe RNA from DNA. So it's going from DNA to RNA. And it only copies one strand. Meanwhile, replication uses DNA polymerase to produce complementary DNA. And it would copy both strands. So transcription is selective. It's regulated by promoters. Replication is semi-conservative, and it occurs once per cell cycle. Again, what's the difference between transcription and replication in terms of enzymes and products? So let's go left and right. Transcription, it would use, well, it is going from DNA to RNA. It would use RNA polymerase, and it is selective. It uses promoters. Meanwhile, replication copies both strands. So it's going from DNA to complementary DNA. It uses DNA polymerase, and what's the next one? It is semi-conservative, and once per cell cycle. Okay, some biochem. Why is ATP a high energy molecule? ATP has high energy phosphoanhydride bonds. Well, that's because ATP, adenosine triphosphate. You can see how there's triphosphate at the end, usually in the pictures. Depending on how you draw them. There is charge repulsion between the phosphate groups. They do not want to be bunched together. They are of the same charge. So that would increase the entropy upon hydrolysis. So when you break the bonds, there is very, very significant free energy released. So why is ATP very high energy? Because of the charge repulsion between its phosphate groups. And how is ATP generated in the mitochondria? This is interesting, because usually we just keep talking about how ATP is used. But here, we are talking about how ATP is made in the mitochondria. Again, what's the mitochondria? It's the powerhouse of the cell. So in oxidative phosphorylation, electrons from NADH and FADH FADH2 would pass through the electron transport chain, ETC. Again, electrons from NADH and FADH2 would pass through the ETC in the inner mitochondrial membrane, pumping the protons into an intermembrane space. And this proton gradient would power the ATP synthase, which synthesizes ATP from ADP and NPI. So in oxidative phosphorylation, the electrons from NADH and FADH2 would pass through the electron transport chain in the inner mitochondrial membrane. And that would lead to the protons being pumped into the intermembrane space. And with that proton gradient, it powers the ATP synthase. ATP synthase would then synthesize the ATP from ADP and PI. What is oxidative phosphorylation and where does it occur? Oxidative phosphorylation is the process of ATP synthase, again, powered by the transfer of electrons to oxygen via the electron transport chain, which would create a protein gradient used by the ATPase. It is in the inner membrane, inner mitochondrial membrane. So what would happen to ATP synthesis if the inner mitochondrial membrane is leaky? That's bad because a leaky membrane would then dissipate the proton gradient, and then that would uncouple the electron transport from ATP synthesis. Oxygen would still be consumed, but ATP production would drop significantly and energy would be lost as heat. So if it is leaky, a leaky membrane would dissipate the proton gradient. Proton gradient would lessen, and thus oxygen would still be consumed. However, ATP production would drop significantly. Again, flow of information from DNA. Okay, this one's new. From DNA to protein, how does that happen? DNA is transcribed into RNA, and then RNA is translated into protein. So DNA is transcribed into pre-mRNA in the nucleus. Pre-mRNA undergoes capping, 5' capping, splicing, and 3' the polyadenylation to form mature mRNA. mRNA is exported to the cytoplasm outside the nucleus, and the mRNA is then translated. Well, that's where the ribosome would translate it into protein using tRNAs and amino acids. So again, DNA to protein. In a nutshell, DNA is transcribed into pre-mRNA, and then that pre-mRNA is then you add a 5' cap, using spliceosome, you splice out the introns and keep the exons, and then the polyadenylation also happens, and now you have a mature mRNA. The mature mRNA is exported outside the nucleus and into the and it's now floating around in the cytoplasm. The ribosome would see the 5' cap and it's like, oh, I have to translate you into the protein, into a certain protein. It would use specific tRNAs and amino acids to make it into the protein. Okay. How does a cell sense and respond to low glucose levels? Interesting. Low glucose, which is what I'm feeling now, I will eat. Low glucose triggers an increase in glucagon secretion. Okay? So low glucose, it will trigger glucagon secretion, which would activate adenylate cyclase in hepatocytes. Adenylate cyclase. So glucagon, so you're hungry, that would trigger low glucose. Low glucose leads to glucagon secretion. Glucagon activates the adenylate cyclase in hepatocytes. What are hepatocytes? Liver cells. And CAMP levels rise, and that would activate pKa. pKa phosphorylates certain enzymes, which would promote glycogenolysis and gluconeogenesis to increase the blood glucose. So that's how the cell senses and responds to low glucose level. It's trying to blood glucose. Why do we need buffers in blood, and what's the main one? What would happen if it failed? Buffers would maintain pH homeostasis, and it's important for enzyme stability and protein stability. And the main buffer is the bicarbonate system, which is carbon dioxide plus water equals CO2 plus H2O becomes H2CO3. And if the system fails, you can have acidosis or alkalosis, which would lead to enzyme dysfunction and altered ion distribution. How does feedback inhibition regulate a metabolic pathway? Feedback inhibition would occur when the end product of a pathway inhibits the upstream enzyme, usually the first step. So example, when you try to make isoleucine or in isoleucine biosynthesis from threonine, isoleucine would inhibit threonine deaminase, which would prevent overproduction of isoleucine. Okay. And I think for now, that is it. Thank you for watching. Bye.